It's time to talk about innate immunity. Remember that all multicellular organisms contain intrinsic mechanisms of defense against infections, which constitute innate immunity. Describe some of the mechanisms of innate immunity, especially how does innate immunity respond to microbes? The mechanisms of innate immunity respond to microbes and not to non-microbial substances, are specific for structures present on various classes of microbes and are mediated by receptors encoded in the germline and are not enhanced by repeat exposures to microbes. That would be adaptive immunity. Describe the basic function of TLRs, or toll-like receptors. TLRs expressed on plasma membranes and in endosomes of many cell types are a major class of innate immune system receptors that recognize different microbial products, including bacterial cell wall constituents and viral nucleic acids. Describe the receptors of the NLR family. Some receptors of the NLR family recognize microbes, products of damaged cells, and other substances, and these receptors signal through the cytosolic multiprotein complex, the inflammasome, to induce secretion of the pro-inflammatory cytokine interleukin-1. What are NOD-like receptors? The nucleotide binding oligomerization domain receptors, in short, NOD-like receptors, NLRs, are intracellular receptors of PAMPs that enter the cell via phagocytosis or pores and DAMPs that are associated with cell stress. They are part of pattern recognition receptors and play key roles in regulation of innate immune response. NLRs can cooperate with toll-like receptors and regulate inflammatory and apoptotic response. They are found in lymphocytes, macrophages, dendritic cells, and also in non-immune cells, for example, in the epithelium. Describe the principal components of innate immunity. The principal components of innate immunity are epithelia, phagocytes, dendritic cells, natural killer cells, cytokines, and plasma proteins, including the proteins of the complement system. How is the epithelia involved in this system? Epithelia provide physical barriers against microbes, produce antibiotics, and contain lymphocytes that may prevent infections. What are the principal phagocytes? The principal phagocytes are neutrophils and monocytes, or macrophages. Remember that monocytes circulate in the blood and macrophages are in the tissues. And remember that these are blood cells that are recruited to the site of infection, where they are activated by engagement of different receptors. The activated macrophages destroy microbes and dead cells and initiate tissue repair. These functions may be carried out by different populations of macrophages. Describe the function of natural killer cells. Natural killer cells kill host cells infected by intracellular microbes and produce the cytokine interferon gamma, which activates macrophages to kill phagocytosed microbes. So here the question you want to remember is, what cytokine is produced by the intracellular microbes? And that is interferon gamma. Sorry, I'm getting confused here. What I mean to say is that the natural killer cells produce the cytokine interferon gamma, which activates macrophages to kill the phagocytosed microbes. Okay, describe the complement system. The complement system is a family of proteins that are activated sequentially on encounter with some microbes and by antibodies in the humoral arm of the adaptive immunity. And complement proteins coat or opsonize microbes for phagocytosis. They stimulate inflammation and lyse microbes. talk about the action of cytokines of the innate immunity. How do they function? The cytokines of the innate immunity function to stimulate inflammation. We're talking about tissue necrosis factor, interleukin-1, and chemokines, or sometimes interleukin-6. Remember that those are the pyrogens, and also the cytokines activate natural killer cells. And remember, this would be interleukin-12, and activate macrophages, as we talked about. This is interferon gamma, and prevent viral infections, which is type 1 interferons. Okay, let's describe the process of inflammation. And before I describe the process of inflammation, I just wanted to mention that the people behind me are not going to be eaten. It's just I have them in the background to add some feeling to my lecture. Anyway, let's continue. Inflammation consists of recruiting phagocytes to sites of infection and tissue damage, a process mediated by binding to endothelial adhesion molecules that are induced by the cytokines, tissue necrosis factor, and interleukin-1, and responding to soluble chemoattractants, including chemokines, complement fragments, and bacterial peptides. This is followed by ingestion and destruction of microbes and damaged cells. Let's talk about the antiviral defense. What type of interferons mediate antiviral defense? The type of interferons that mediate antiviral defense are type 1 interferons, which inhibit viral replication, and NK cells, which kill infected cells. In addition to providing early defense against infections, innate immune responses provide signals that work together with antigens to activate B and T lymphocytes. The recruitment for these second signals ensures that adaptive immunity is elicited by microbes, the inducers of innate immunity reactions, and not by non-microbial substances. Thank you very much for listening to this lecture, and I hope that you tune in next time. Same shark time, same shark channel, although there may be another animal or another character giving a lecture. Thank you very much, and let's enjoy immunology.